Specialized crystal forming cells and crystal chambers. Number 1 is calcium oxalate crystallization in specialized plant cell. In higher plants, calcium oxalate crystals typically form inside the vacuole of specialized cell. Some studies have suggested that excess apoplastic calcium induces differentiation of these cells, but other research has shown that they differentiate even when calcium supply is limited. A large body of ultrastructural research documents the unusual development and unique feature of crystal forming cell in variety of plants. However, detailed correlative information about the physiological and biochemical features that characterize these cells is generally lacking, because structural and developmental features of crystal forming cells in plants have been extensively reviewed. In most biomineralization process, specialized cells and organic macromolecule in or around specialized cells govern and mediate crystal formation. These cells and their associated molecule collectively turn the organic matrix function in a variety of ways to compartmentalize the crystallization process, to nucleate crystal, and to modify crystal growth and morphology. Second one is crystal formation within intravacuolar membrane chamber. Although structural features of crystal forming cells vary in relation to crystal morphologies in diverse plant species, certain characteristics are common to most systems. For example, crystals typically develop inside the vacuole. They form within intravacuolar membrane chamber termed crystal chamber that differentiate and proliferate exclusively in the vacuoles of crystal cell. Additional substances commonly accumulate specifically within the vacuole of these cells, including paracrystalline bodies, tubular or fibrillar structure, and various densely staining substances that associated with the crystal chamber. Mucilage consisting mainly of complex polysaccharide also may accumulate within such vacuole, particularly in association with raphides. In certain plants, paracrystalline bodies or tubules appear to give rise to crystal chamber. In many other plants, the origin of crystal chamber has been established. Now, the question is, how are calcium and oxalate ions transported to crystal chambers? Because crystal chambers compartmentalize the crystallization process within the vacuole, it is logical to conclude that they provide selectivity and control over entry of ion into the crystallization space. However, there is no information about transport proteins in this membrane, whether or not accumulation of calcium and oxalate in the chambers require energy depends on mechanism of transport. The second question is, what is the source of oxalate for crystallization? The source of the oxalate that must be synthesized in the substantial amount required for crystal formation is unknown. In certain plants, oxalate synthesis increases with increased calcium supply and uptake.